Hello children, am I visible? Am I audible? Done? Yes. Okay. So I think, I'm so sorry, there was a technical glitch, which is why it happened. So we are back again. Let's wait for a minute so that everyone else gets to join, those who were there in the previous live. Okay, I'm so sorry. There was some Wi-Fi fluctuation that happened. So the rest of the questions we will be solving now. Okay, so children who have subscribed to the channel will only be able to put down your answers or ask me doubts in the live chat. So make sure you're doing that. Okay, now going ahead to the next question that we have, and that is this. Okay, hi dear, hi Nagraj. Okay, so identify A, B, C, D in the following table. That means papaver somniferum, what drug does it form? And this drug has to be a depressant. So it would be heroin over here. So answer for this question would be heroin. Similar type of question we solved, okay, while we were doing MCQ questions. Now cannabis sativa, over here the drug is cannabinoids, and what do? They affect the cardiovascular system. So that has to be written. So cardiovascular system. Cool. Next question. Erythroxylum coca. What is a drug that you get? You get cocaine. You get cocaine. And what does it do? It interferes in the dopamine transmission. It interferes in the dopamine transmission, and also you can mention about CNS. Okay, central nervous system. Either of them, if at all you're right, you get marks. Fine. So these are the things that you need to write for 27th question. If at all you have written something else, make sure you're going through it once more. Check whether your answer was correct or not. Okay, cool. Let's go ahead with the next question out there. These are 3 marker questions, and you have to attempt 5 of them. Okay, what is artificial hybridization? Name and define the techniques that are employed to achieve this. So you have to mention about emasculation. You talk about emasculation, what happens, you remove the stamens, and then you talk about basically bagging, and then you talk about pollination, artificial pollination, and then you talk about rebagging. So all these things are really important. Ma'am, for 26th answer. So 26th answer was 19, dear. You are asked about cytosine, the answer would be 19. Okay, we solved it. Fine. So 27, 28. So we are over here. I hope you know what all you need to write. Now what is menstrual cycle? This is the reproductive cycle in the case of human females. Okay, and then you talk about its different phases out there. What are different phases? First you have menstrual phase, then you have follicular phase. What is the other name for follicular phase? You call this proliferative phase. And then the third phase is luteal phase. You also call this as secretory phase. In between them comes what? Ovulatory phase. We can talk about that as well. So these are the 4 things that you need to write: menstrual phase, follicular phase, the other name of it, and ovulatory phase, and the last phase is luteal phase. For all of them, whatever parallel names are there, write them down as well. Cool. Now going ahead with the next question. ZIFT is a boon to childless couple. Explain the procedure. So what is ZIFT? First of all, you have to explain the term ZIFT. ZIFT stands for zygote intrafallopian transfer, and then you have to mention this is a process where what is happening, either you put the zygote which has been made in the laboratory condition into the fallopian tube of a female who is not able to naturally conceive, or you either put embryo up to 8 blastomere stage, not more than that, and then it will follow its regular development. But here you have to mention fertilization is happening outside the female body. Mention all this small things out there. Okay, and this is part of what? Assisted reproductive technology. Cool. Next, a child has blood group O. If the father has blood group A and the mother has blood group B, work out the genotypes of the parents and the genotype of the other offsprings. So you have to talk about all the siblings. So it is mentioned that the child has blood group O. Do you know when does O come? From both the parents you should get at least 1 IO. At least 1 IO, and here it is mentioned the mother is A, sorry, the father is A, and the the mother is B. So for a father to be A, the blood group can be like this, where A is dominant over O. Similarly, a mother to be B, the blood group can be this, where the B is dominant over O. So this would be what? This would be the genotypes of the parent. Now you have to talk about the siblings. What are their genotypes? So write it properly, IAIO, and here IBIO. Let's see. So if you cross this and this, you get IAIB. This is the genotype. What is the phenotype? AB blood group. When you cross this with this, you get IAIO. Genotype is this, phenotype is A blood group because A is dominant over O. Fine. And over here, this IBIO. Here B blood group because B is dominant over O. And lastly, you have IOIO, that is O blood group. So talk about it in detail. Write parents, write the gametes, write the genotype. That's how you need to go. Cool. Now going ahead with the next question. Children, you can stop me if at all you have any doubts. Okay. At some point, if at all you have any doubts, just ask me. Now going ahead, yes, A, B, A and O. Yeah, exactly. A, B, B, A and O. O is the, what do you call, the child, the blood group of the child, right? So yes, rest of it you have to write. Next question, you have to talk about RNA polymerase exhibit clear-cut division of labor. Justify the statement. What do you mean by that? I told you again in the short video it was there. There are 3 types of RNA polymerases, 3 of them doing different jobs. RNA polymerase 1 transcribes rRNA, that is ribosomal RNA. RNA polymerase 2 transcribes hnRNA and mRNA. RNA polymerase 3 transcribes what? It transcribes tRNA, transfer RNA, and 5S rRNA. So the 3 RNA polymerases which is there, and what are the jobs they do, you have to talk about that over here in this particular question, 32nd question. Okay. So there are 3 types of RNAs which each functions, write them. Yes, Nagaraj. Next, draw a neatly labeled diagram of S.L. Miller's experimental setup. You draw it, you mention about all the conditions. Ma'am, for 30th question, for writing full form. Okay, 30th question. What is the 30th question? ZIFT. You have to write the full form. Yes. Zygote intrafallopian transfer. Okay. Now over here we were doing, yeah. So here you have to mention about the conditions. For example, what all things were there, such as methane, such as ammonia, water, and also H2. So all these things should be given. 1 marks they will give or not. 30th question. 1 marks for a 3-marker question. 1 mark, I think, yeah, 1 mark or half a mark, definitely you might get it. Okay. But I think there is a little bit of explanation needed. Hopefully, if the teacher is very lenient, you may get it. But mostly you should be explaining at least the definition or, yeah, the full form very nicely with something else as well. But we can expect 1 mark out there. Don't worry. Let's think for good things. Okay. Meenakshi? Now, what is cancer? Differentiate between benign tumor and malignant tumor. So, what is cancer, my dear children? Where there is uncontrolled growth of cells, and these cells basically move around to different positions. They show metastasis. This is known as cancerous cell, and this disease is known as cancer. Now, over here you have to talk about benign tumor and malignant tumor. Benign tumor is made up of cells. Okay, they show uncontrolled growth, but they are restricted to one place. They don't show metastasis. On the other hand, malignant tumor is caused when the cells show metastasis. They move to different areas of a body and they get localized over there. So that is the thing. So benign tumor is a local tumor. Malignant tumor is the dangerous version of tumor where the cells are cancerous. They move to different areas by a process that is known as metastasis. So you have to mention about that. Cool. Now, write any 4 questions in 5 markers. So first is name any 4 devices that have been developed in flowering plants to discourage self-pollination. That means you have been asked about outbreeding devices. So you have to tell. First is what we discussed it so many times. First one, the position of anther and stigma. It can be a different position. Suppose anther is here, stigma is there. So obviously this will prevent self-pollination. Second thing is basically maturation time. Different maturation time of stigma and anther. So stigma might be receptive, but anther might not be. So that can be happening. The third one is basically self-incompatibility, a genetic mechanism where even though pollen grains land on the stigma, still they won't be able to form the pollen tube. So that can also be there. Okay, and the fourth thing, formation of unisexual flowers. If the flower which is there, it does not have both the sexes, then to some extent you can stop what self-pollination to happen. There will be always cross-pollination in that case. Okay, now cross-pollination, true cross, either gytenogamy or xenogamy, both of them can be possible depending on the plant. So all these 4 things you have to mention. First, talk about outbreeding devices. Here they have not mentioned outbreeding devices, so you have to talk about that this is known as outbreeding devices. They prevent inbreeding depression, and then start off your answer. Now what is seed dormancy? When the state is dormant state, it is viable, but it is waiting for the conditions to what? Germinate. So as of now, it's not germinating, but it's in the condition where its metabolic rate, everything is low. Okay. Now name the plants which show 2000 years and 10,000 years of dormancy. So 2000 years plant is what? Phoenix dactylifera. 10,000 years is what you call Lupinus articus. So these two things are there. It is given in your NCERT in sexual reproduction in flowering plants. Go through it. Okay. So over here, this is Phoenix dactylifera and that is Lupinus articus. Go. Now, draw the diagram of human sperm and mention all these things. So you have to draw the diagram of that. Now, yes, nothing else. Write the schematic representation of dihybrid cross. So over there, you have to talk about the whole dihybrid cross. Ma'am, is this important for every district midterm question paper? So, my dear child, I'll say Sumit, hi, first of all. And second thing, listen, the questions will not be repeated as such. It's the concepts which are repeated. Okay, so you should be more sure about the concepts. Now, some people might say the question will be repeated as such. It's totally false. Do not go behind their words. Okay, so I had made a separate video on short concepts. Okay, you can go and check our channel. It was released yesterday itself. In that, the concept is there and the type of questions that are framed from these concepts. So, please make sure you're practicing those concepts. Those are the most repeated ones. Okay, that can be told. Now over here, obviously so many questions are from those concepts. So yes, you can refer to that video. Now, yes. Now, yeah, so you have to talk about the dihybrid cross experiment fully, make the Punnett square, talk about the phenotypic ratio, that is 9:3:3:1. What does 9 stand for? What does 3 stand for? All of it has to be mentioned clearly. Okay, because over here it is written phenotypic ratio. So what does 9 stand for? Everything clear. Next, write a note on the cause and inheritance of color blindness. So in that, you have to talk about color blindness, different scenarios you can explain, where the mother is carrier and the father is color blind. Okay, you can talk about that, and you have to mention that it is what? A sex-linked recessive disorder where the cone cells are affected. Sorry, certain red and the red color and the green color cells are being affected. You have to talk about that. Okay, ma'am, when does the Sankalp batch continue? We are doing it really soon. Okay, this month end you can find it. So yes, so talk about different scenarios. Okay, where the mother is a carrier, where a mother is a carrier for the disease and the father is normal, or when the father is having the disease and the mother is a carrier, or when the father, just a second, okay, or when the father is having the disease and the mother is also having the disease. So all about these scenarios we can explain again with proper things. Ma'am, for 35A, they allotted is 2 marks, then only 4 name is enough or not? 35, where is 35? 4 names, yes. 4 name and explanation, my dear children. Little bit of explanation would be enough. You have to explain. Name any 4 devices which has developed. So please explain a bit, but since it is written as name, it would be enough. But I definitely say in every examination, whether this or boards, my dear children, if at all, okay, if at all, name and this thing is written, explain a bit of things. It's very important for you. Okay, Shiloh's homely food. So my dear child, I had discussed the MCQ questions before. Okay, in a previous live, so that live got stuck automatically. I could not explain. So over here I am going ahead with this. Okay, if you want I can explain it to you once more. Cool. Now, how does thalassemia differ from sickle cell anemia? You have to explain that how. Because thalassemia is a quantitative disorder, sickle cell anemia is a qualitative disorder. Both of them are blood disorders, but then explain them separately. Why it is quantitative and why is it qualitative? Because over here the quality of beta hemoglobin chain is being hampered. Why? Because there is a point mutation happening at 6th position of the codon. All of this thing. In thalassemia, why is it a quantitative disorder? You have to explain that in alpha thalassemia these genes are there, but if one is affected, still alpha globin chain will be produced, but not too much. Okay, so this is of Bangalore. Okay, Sumit, this is of Bangalore South Zone. Fine. Now, yes, so these are the things that you need to explain. Now, list out the salient features of human genome, all the features which are there, that it has 3,164.8 million base pairs, and then 30,000 genes are there. Some bases, usually the average size of a gene is 3,000 base pairs, but in dystrophin the case is different. All of it you have to explain line by line. Okay, in chromosome number 1 has these many genes, chromosome number Y has just 231, whatever points were there in the human genome, you have to mention that. Now, 40th question, mention the different stages of human evolution in correct sequence. So you have to start with what? Ramapithecus and Dryopithecus. Say that Ramapithecus were more man-like, Dryopithecus was ape-like, and then talk about Australopithecus, and then talk about Homo habilis, Homo erectus, Neanderthal man, and then lastly you have Homo sapiens. So those are the different stages you have to mention like that. Okay. Now, what are the homologous and analogous organs? Say that homologous organs, basically they are structurally similar, but they are since of adaptations to different habitat, they would be doing different functions out there, and they show divergent evolution. Give example of what? Of forelimbs of human beings, whales, and what do you call, bat, and all those things. All of them are going to use it in different ways. Analogous organs, where basically structurally they are not similar, but functionally they show convergent, they result of convergent evolution. Give example of what? Butterfly wing and bird's wing. All these things can be explained. Okay, give an example of each is mentioned. Even if it's not mentioned, please explain. So as I always say this thing, in question, whatever is asked, go a little extra and talk about it, but not too much so that it wastes your time. Bit extra, not too much, okay? Now, what is innate immunity? Mention the 4 barriers of innate immunity with an example for each. You have to talk about them. So, physiological barrier, physical barrier, cytokine barrier, okay, cellular barrier, the 4 things which are there, you have to mention about it. Explain innate immunity, that it is non-specific time of immunity present with us from the point of birth, all of these things, okay. So as I said, these questions are something that have been repeated. Okay, they have been repeated from the short. The concepts are so much repeated. So my dear children, watch the short video if at all your exams are coming by. Okay, and then practice. Now over here, you have to pick one 5 marker. Draw a diagrammatic sectional view of male anatropous ovule. So anatropous ovule means ulta ovule. So over here what will happen? Usually the placenta and the funicle is over here in a normal ovule. But in this case, the placenta and funicle would be at this particular site rather than here. Okay, and then you have to mention about everything else, that they would be having integuments, there is chalazal end, there is micropylar end, there will be cells of nucleus, all of it. So that is anatropous ovule. So, and you have to mention what? That develops into seed coat. What is that something that develops into seed coat? It is the integument. So mention about the integuments. The next thing, so this is integument. Next, that attached to the ovule to the placenta, this is known as funicle. It is funicle. And next part, that develops into embryo after fertilization. That develops into the embryo after fertilization. It would be what? So you have to mention about the embryo sac, and then you can say that basically there is an egg cell, and the egg cell is the one that develops into the embryo sac. Okay, Meenakshi, example for 40th B, limbs of birds and potato is correct. So potato and sweet potato, okay, limbs of birds, mammal, I'm so sorry, limbs of man, whale and bat and cheetah, all of them are examples of them, are examples of divergent evolution. A simple category, that is mammals, but all of them are diverging into different points. So that is the thing. Limbs of birds cannot be an example. Okay. Now for the next thing, convergent evolution, you can talk about potato and sweet potato example, or you can talk about the wings. Okay, the wings of birds and butterfly. Fine. Now coming over here, okay, that's okay. The basal part of the ovule, what do you call the basal part? It's known as chalazal end. And now the cells which has abundant reserve food materials, it is known as nucellus. It is known as nucellus. Okay. Which paper, ma'am? So this is the paper for South Zone of Bangalore. The exam had happened today, my dear children. Cool. Fine. Now, given below is the sequence of nucleotides in a particular mRNA and amino acid coded by it. So, UAA, AUG, UUC and everything is given. Write the properties of genetic code that can be and that cannot be correlated from the given data. That means you have to talk about everything, all the features of genetic code. So, UUU stands for phenyl. That means it is an unambiguous. Okay, it is what? Unambiguous. Each codon is coding for one particular amino acid. Then talk about it is comma-less. Okay, then say that it is triplet codon. Okay, and so first you talk about the things that can be excluded from this. Then you talk about the things that you cannot. So, for example, you cannot say that okay, this is degenerate feature as well. This is degenerate feature of the codons, and over here you have the stop codon. Start codon has not been mentioned, so you can mention that separately. So AUG is the start codon, something that is not been correlated over here. So explain that separately. So here you can find unambiguous nature, degenerate nature, comma-less, triplet codon, the stop codons, everything. What you cannot find is what you call AUG is not there, the start codon is not there. Okay, and it does not show the universal nature as well. Oh, so AUG is over here. Yeah. Sorry. So AUG is over here. So yeah, you don't have to explain. You have to explain the correlated one. In the non-correlated one, I think universal thing can be mentioned. Okay, that the amino acids, the codons which are there, they are universal. So that cannot be seen up over here, right? Yes. Now, why does a doctor administer tetanus antitoxin and not a tetanus vaccine to a person injured in an accident with a bleeding wound? Justify. So please mention exactly what you find in tetanus antitoxin. What is this type of immunization? This is known as passive immunization, where you are giving ready-made antibodies such that the person can be safe. If you are giving tetanus vaccine, you are going to give live attenuated form of the pathogen, and you will wait for your own body to produce antibodies. That is active immunization. So do not want that to happen. You want passive immunization. You are giving ready-made antibodies so that the person can be safe as soon as possible. Okay, cool. So that can be done, and that's what you have to tell. Now, a young boy, when brought a pet dog home, started to complain of watery eyes and running nose. The symptoms disappeared when the boy was kept away from the pet. Name the type of antibody and the chemicals responsible for such things. So antibody is IgE for allergy. Okay, IgE for allergy, and the chemicals responsible for it is that the mast cells onto which these antibodies are there. Okay, so antibody is here, mast cell is there. So whenever an antibody gets a signal from an antigen comes and binds to the antibody, the cell becomes active and it is going to release two things, serotonin and histamine. Serotonin and histamine. So these are the two chemicals which are there for the response. Now mention the name of any two drugs that could be given to the boy for immediate relief from such a response. So what is the name of the drug? In your NCERT it is given. It is antihistamine, and the second thing is adrenaline. Okay, antihistamine, and also adrenaline, and then you can give steroids as well. So these are few things that you can write. Two of them are being asked, so you can just write two. That would be enough. Cool. Now, for visually challenged students, there are certain questions which are the following: secretes corpus luteum. It would be Graafian follicle that secretes the corpus luteum. Which of the following is an example for barrier method? Condom is the example for the barrier method. How many base pairs are present in a typical nucleosome? Okay, it's 200 base pairs. And then what is pedigree analysis? Write its significance. You have to talk about significance of that, and describe any 5 characteristics of genetic code. So as I said, unambiguous and all those things you have to mention. So these are the things. That's done, my dear children. With this we end. If there is any question that you need to ask, you can ask me right away. And my dear children, for getting all the understanding of everything up there, do not forget. Okay, that was a thunderstorm that came inside also, I think. Yes, so if at all you have any doubts, as I said, do not forget to ask me, and do not forget to click on the subscribe button right now. So my dear children, I think I'll end. It's raining so badly here, in Bangalore. So as of now, wishing you best for the next examination that you are having. And as I said, if at all you have any doubts, put it in the comment section below. I'll read it later on, and watch the short video. It is going to help you really well. Shailus, ma'am, biology is feeling very tough. What should I do? So biology becomes easier when you study it every single day. If at all, so when it piles up, my dear child, what happens is that we are like, oh my god, so much things are left to study. So make sure you're watching at least one concept video or anything like that every single day. Have that streak, and then it will automatically become easy. And when you do a concept and do a question, it will be more easier for you to, you will feel confident that okay, I am able to solve the questions as well. Fine. So that's how we can tackle it. There's no other shortcut to liking biology or feeling that it is going to be easy. Actually, it's an easy subject, but it becomes difficult of its huge syllabus. Okay, so many things to remember. So have the habit of understanding the concept each day. Have the habit of recalling whatever you have studied by yourself, not reading the NCERT, just recall it by yourself. Okay, and solve the questions. That's all. Okay, Farhan is asking, ma'am, can I go to washroom, please? Okay, so, ma'am, English paper discussion not done. Why, ma'am? So there was some problem. We recorded the video, but the video got corrupted, which is why the video was not delivered. And yes. So yes, so I'll wind up this live and wishing you all the best, my dear children, for your coming examination. And if at all you do not have exam, go sleep. Okay, so go sleep for some time. Have a good day. Wishing you all the best. Lots and lots of love from Jasmine ma'am. And as I said, stay tuned by subscribing the channel. There are so many things coming up for you guys. That's all. Okay, so bye bye. Take care. Lots of love. See you soon.